Hello everyone, my name is Moises, and today I will be showing you how to grow an overnight culture of E. coli cells with flavin protein plasmids. First, we're going to provide a little bit of background of what the procedure is going to be. Growing an overnight culture is a simple but crucial step in biochemical research. This procedure is usually carried out in order to prepare cells for another experiment. Examples of this are to prepare an inoculum for a larger culture or to simply generate a sample of cells from which to purify plasmid DNA. Generally, this procedure serves as a day one of a larger multi-day experiment. In this particular video, we are going to demonstrate how to properly inoculate a 5 milliliter starter culture using appropriate antibiotics and a single colony of E. coli, which contains the desired flavor protein plasmids. This will then be grown with shaking under 220 RPM at 37 degrees Celsius and left overnight. It is important that at all times we maintain aseptic technique as materials can become contaminated by exposure to air. This includes, but is not limited to, keeping things closed at all times unless in use, as well as using a flame sterilizing method via a Bunsen burner both before and after anything is removed or added.
To begin the procedure, you will need to get a bottle of pre-autoclaved Luria Bertani medium, a sterile culture tube, 10 milliliter sterile serological pipette, a 10 microliter pipette, and a plate in which you will use to inoculate your culture. To begin, without fully exposing it to air, attach the 10 milliliter pipette to the pipetter. Then, we light the Bunsen burner and open the bottle of medium. We loosen the cap of the culture tube without removing it. This is to prevent any sort of contamination. The next step is to pipette 5 milliliters of media into the culture tube using sterile technique. Once the antibiotic is thawed, we add 1 microliter per every 1,000 microliters of the culture. In this case, 5 milliliters of culture is being used. Therefore, we add 5 microliters of our antibiotic. Using a sterile tip and sterile technique, we transfer our cells from a single colony onto a plate in order to grow our overnight culture. We spread it evenly using spreading techniques,
Finally, using a sterile tip and sterile technique, we transfer cells from a single colony from your plate to your overnight culture tube. We then place the culture in the incubator and grow overnight with shaking at 220 RPM and 37 degrees Celsius.